Okay. Let's pick up where we left off. And last time we were talking about genetics. So we left off here. I'm going to go ahead and just remind you that if you have any questions or anything, to be sure to put them in the chat box anytime throughout lecture. All right. So last time we met, last Thursday, we had finished talking about protein synthesis, which is really exciting. So we know the basics of how we go from DNA to RNA to proteins. So just a reminder, when we're talking about that process, what we're doing is we're going from a genotype, which is the set of genes you have, so DNA, and making a copy of that in the form of mRNA, and then getting that mRNA to a ribosome, and then that's where that mRNA is made into a protein. So this is basically how we go from all the instructions of life found in your nuclei or the genome of organisms to kind of the physical manifestation, how we go from genotype to phenotype. So I was looking at the discussion board, and one of a kind of a common question I had that I noticed was about introns and exons. And I just wanted to briefly go over this. I'm going to go through the discussion board as well to kind of go over those questions as soon as I can. My goal is hopefully to do that today, but so I can more specifically see what it is about exons and introns. But I just wanted to briefly mention that introns and exons are both present in DNA and what we call pre-mRNA. So that's what we're kind of looking at here. Sometimes this mRNA that is transcribed from the initial DNA before it goes through processing is called pre-mRNA. And that's because it still contains the transcript of both exons and introns. So you can see here what we have in our DNA. We have all nucleotides, right? So remember, DNA is made of nucleotides, A, T, C's, and G's. And it's a sequence of those nucleotides. And that's a gene. But within that nucleotide sequence, there is varying segments. Some of those segments are exons, and some of those segments are introns. So the difference here is that exons are segments of nucleotides that will directly contribute to codons, which, remember, codons are what code for amino acids in a protein. So you can kind of equate anything in an exon to something that is going to eventually be translated. However, introns are segments of nucleotides that we're going to cut out. And the reason we cut them out after we generate our pre-mRNA, when we go through processing, is because introns are segments of nucleotides in the DNA, and those nucleotides do not directly contribute to amino acid sequence, right? So they're nucleotides, but we don't want those sequences of nucleotides being transcribed into codons. They will not contribute to the overall phenotype. So what we do is we have these segments, exons and introns. So remember, the exons are nucleotides that will be transcribed eventually into coding segments, so portions of the mRNA that will directly contribute to translation of protein. Whereas introns are segments of DNA as well as pre-mRNA that does not contribute to codons and therefore protein. So what we have to do is, and if you're kind of like, what's the purpose then? Like, why? That's what we're going to talk about partly today. So we will get into that. One of the reasons is to kind of like add spacers, or historically that's one of the hypotheses, is that it adds spacers to the genome. So if there's any, there are so many introns, like I'll talk about later, like even though it seems like a ton of your DNA would need to be exons, so contribute to protein, the vast majority of your DNA is actually non-coding or intronic. And one of the original hypotheses was that they functioned as spacers. So if the majority of your genome is introns, that means that you can get mutations throughout your DNA that won't disrupt the eventual production of proteins. So kind of just spacers. As we'll talk about today, once we move into it, those segments of DNA are actually really important for other reasons as well. But yes, so I had a great question. Do introns contribute to regulation at all? Absolutely. And we'll get into that. That's kind of what we're going to talk about today. So originally, one of the hypotheses was that they contribute mostly to just being spacers. What we know now is that those intronic sequences don't necessarily contribute to a phenotype, but what they do is they contribute to regulation of transcription and translation. So we'll talk about that later today. Okay, so we'll talk about introns, but it is really important to note that one of the big differences here is that in your DNA, and as well as that in your pre-mRNA, what you have a sequence of nucleotides that is divided into exons and introns. Only the exons, or only the nucleotides that are in those exons, will eventually contribute to codon sequence and therefore protein or amino acid sequence. So what we have to do is we have to transcribe DNA into pre-mRNA, both exons and introns. And then what we do is we process that mRNA. One of the things we do during processing is we splice out those introns. The reason we splice out those introns is, again, because they do not contribute to the amino acid sequence. So we just don't need them in there. Again, historically it was like, well, if they're spliced out, they're not important. They're very important, but they're very important in regulating transcription and translation. And by the time that we are actively translating, then they're kind of like irrelevant. So a lot of their function as far as regulatory segments happens before translation, as we'll talk about. All right, so I hope we kind of answered the difference between introns and exons, but again, I'll go through that discussion board and talk a little bit more about it on there as well. So that is one thing I noticed a question. And what I wanted to do today to sort of review where we left off is just, this is an overall view of the entire process we talked about in last week, which is we start with DNA. Okay, we have double-stranded DNA. And remember that the strand of DNA that will be transcribed into mRNA is the 3 to 5 prime strand. So this is called the template strand because it serves as a template for the creation of mRNA. So that DNA strand is in the 3 to 5 prime direction. It's transcribed by RNA polymerase, and that results in the production of a 5 to 3 prime strand of mRNA. So this mRNA is complementary to our DNA strand, remember? So if you have a T in your DNA strand, the complement to T is A. If you have a G, C, A, because it's RNA, that would be U. And then we go all the way. So that's mediated by tRNA that's by RNA polymerase. And then what we have to do is what we talked about on Thursday is take that mRNA, and what happens first is it's like spliced and processed. But then we know that our mRNA is divided into 3 nucleotide long segments called codons. And those codons code for tRNA, which carry amino acids. So remember that the start codon is always, 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 always AUG, and that codes for our very first amino acid, which is methionine or MET. And then we just go codon by codon. AUG codes for met, AGC codes for serine, and we can always see that based on the codon table. So I just want to remind you what you should focus on when you're doing transcription and translation is the template strand of DNA, 3 to 5 prime strand of DNA, which creates a 5 to 3 prime strand of mRNA. And then codons are in mRNA. So the reason I say that, and we'll talk about this in a minute, is this table is talking about the mRNA strand. So one of the points for confusion, notoriously, that gets students is they think that this table has something to do with like anticodons. Don't even like think about anticodons, really. The only thing you should know about anticodons is that anticodons are how we get tRNA to recognize the mRNA strand. So for example, because everything in genetics has to do with complementary base pairs, you can see if you have a codon being AUG, the tRNA carrying methionine recognizes AUG because its anticodon is complementary to this codon. So A, U, U, A, G, C. It only has to do with recognition. But when we talk about mapping from DNA to RNA to amino acids, we read the codon table based on the mRNA, the codons. Okay, so what I wanted to do, and what I was hoping you would do, hopefully everybody can see this, my Word document, is I wanted, oh, I have a question. How does something like radiation or smoking alter nucleotides and cause mutations? We will talk about that. So we're going to talk about that probably not today, but we're going to talk about that Thursday. So today I'm hoping to talk about genetic regulation, so how we regulate transcription and translation. And on Thursday, we're probably going to focus on genetic mutation. So we'll talk about it, I promise. I guess, yeah, we'll talk about it. Okay, so what I want to do for this slide is please note that what we were given is this DNA strand. So we were given our DNA is 3' to 5'. I'm actually gonna, if I can, sorry, redo that. There we go, just to make it compatible. And file, oops, select all. Okay, there we go. I know that's weird, but I promise it makes sense in a minute. Okay, so what I want us to do is, we know we are given a DNA strand, and you should always be given what direction that DNA strand is. So this slide is giving us a DNA strand in the 3' to 5' direction. So that's very important because that means that it is giving us the template strand. So if you were given DNA that was in the opposite direction, 5 to 3, the first thing you would need to do is use your knowledge of complementary base pairing to figure out what the template strand code would be. So remember that if you're given a 5 to 3' strand of DNA, you need the 3 to 5' strand of DNA. That is your template strand for transcription. So luckily we're given the 3 to 5' strand. So we know the first thing that we need to do to build a protein is make our mRNA, and our RNA is always in the 5' to 3' direction. So what we do is we just go nucleotide to nucleotide. So we know that the complement to T is A, the complement to A is in RNA, U, right? And then CG, T, A, U, C, 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 T is A, G, A. Correct me if I'm wrong, because sometimes I do get this wrong. T, A, C, U, 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 A. Okay, and then of course that is 5 to 3. If you don't have them memorized, can you find them in the chart? So here's what you need to memorize. You do need to memorize the complementary bases. So for example, A to T in DNA, or U to A in RNA, and C to G in both DNA and RNA. But you do not have to memorize the codon table. So you do need to know complementary base pairing. So for example, you do need to be able to take a DNA strand and build a complementary RNA strand, knowing that the complement to T is an A. In DNA, the complement to A is a T, but in RNA, the complement to A is U. You do need to know those complementary base pairs. But you definitely do not need to memorize the codon table. That will be a class way further on than this one, if you ever have to do it. Okay, so we have our mRNA strand now, and we know that our RNA is AUG, AAU. It helps me to kind of separate this into 3 nucleotide segments like I did, again, because codons are 3 nucleotide segments. So now we have our RNA, and what we can do is we can build our amino acid strand. All right, so our first amino acid we see is our first codon, sorry, is AUG. So we find that in our table, AUG, and that's our start codon. Great. All right, so we know that this is met. I'm gonna get the spacing right here, but I probably won't. Okay, and then our second codon, AAU, we go over here, AAU is asparagine or ASN. Okay, our third codon is CCC. We find C, which is proline. AGA, which is arginine. And then we have CAG, which is glutamine. And then you have CUU right here, leucine. And then UAA, which is a stop codon. So it's important to note here, stop codons, you don't need to write stop or anything like that because it's just not an amino acid. So the terminus of this polypeptide would just be the leucine. Okay, so here's another thing that I want to point out. So if you were, let's kind of do another example. So again, if you were given a DNA strand that was in the 5 to 3' direction, the first thing you would need to do is make a complementary DNA strand in the 3 to 5' direction, because that is the template strand. 5 to 3 is our coding strand, which we don't use for transcription. We have to have the template strand. And then we use complementary base pairing to create a 5 to 3' strand of mRNA. All right, and then we can do our amino acid sequence. But the big thing I wanted to point out here, so using codon table. All DNA to DNA, DNA to RNA, that's all complementary nucleotides. And then the amino acid sequence is using the codon table. So again, I will provide you with the codon table, but you do need to know those complementary base pairs. So one thing I want to mention is, let's say that your RNA strand was, let's say it was CUC, CCA, CAA, AUG, AU, CCC, UAA. Okay, so let's say you had this as your amino acid strand. Something that is very important here is, remember, translation does not start until you get to that start codon. So if we look at an RNA strand, we go through this whole process and we get an RNA strand, and you see CUC codes for leucine. That's not our start codon. Remember, our start codon codes for methionine. That will always be the start of translation. So if you have an RNA strand, this is not a start codon, okay? CCA is proline. That's not a start codon. CAA is glutamine, not a start codon. And then we finally get to AUG. Translation does not start until we get to AUG. We know AUG is methionine, and then AAU, I think I used just like, I guess I did use different ones. AAU, asparagine, CCC, proline, and then UAA is a stop codon. If you were given an mRNA strand, it doesn't matter if there are codons that come before that start codon. Translation does not start until the start codon. So basically you just kind of like ignore those others. What happens to the first three? Usually, so you can kind of see this, it's a good question. It's sort of here if you, oh my gosh, this is in my way, sorry. If you see this strand of mRNA, you can see that there's ACU, GCCC. The first couple of nucleotides in the mRNA just contribute to that 5' cap. So they are just untranslated. So you do not start translation until you get to that start codon. It's an excellent question. So yeah, let's see if I can do this. Insert, shape. There we go. Those would be translated into a 5' cap. Okay, so another thing is sometimes you will see at the end of this UAA, you will see AAA, etc. I'm just going to do that for now. What you're looking at that would be your poly A tail. So you'll always have on the end here, this has stop codon and then an untranslated region in the PowerPoint, but that untranslated region will sometimes have other nucleotides in it as well, but it'll primarily be, it's called a poly A tail because mostly it has a ton of A's. All right. Okay, so that's how you would go from, where is this? That's how you would go, this is what I'm looking for, from DNA to RNA to protein. So those are the really important things to know there. You need to be able to do complementary base pairing. You need to be aware of the fact that it's the template strand of DNA, the 3' to 5' strand of DNA that is used to create the mRNA. mRNA is always in the 5' to 3' direction. And then for going from codons to amino acids, you always start at the start codon and you only go until a stop codon. So again, our stop codon was UAA. After that, you do have AAA, right? But which codes for lysine, that is a codon, But because you got that stop codon, that is where translation stops. We do not translate anything after a stop codon. Right. So, all right. Read something about a Tata box. We're going to talk about the Tata box right now. Well, soon. Good question. Okay, so does everybody feel comfortable with kind of DNA to RNA to amino acids? That is something you will have to do on exams coming up. So it is important that you can do that. I will post this to Canvas as well, so you can see it as a reference. All right. Okay. So let's go ahead. Oh, can you explain that stop you mentioned? Yes, yes, yes. So again, if you're looking at your RNA strand, right, you don't start translation until you get to the start codon. Start codon is always AUG. You translate, these moved around, I think, because I moved that. And because of this. Let's see if I can make this a little bit more obvious. But you translate the codons. So for example, your start codon is AUG, and then AAU, CCC, those are codons that code for amino acids. But when we got to UAA, if you look at the codon table, UAA codes for a stop codon. So if we look at what happens when we reach a stop codon, if I can go back there, when we reach a stop codons do not code for amino acids. They code for what's called release factor. And what happens when release factor binds to a stop codon is it just disassembles everything. So what that means is if we get to a stop codon, UAA is one example of those stop codons. If we get to a stop codon, that means that at this codon, release factor will bind and the whole thing disassembles. So that means that even if there are nucleotides following that stop codon, it doesn't matter. Those are not translated. So yeah, I hope that makes sense. Good question. Okay, so let's move forward. This slide was just meant to kind of give you an idea of what we just talked about. Again, I will also post that Word document that I just created for you as well. So we do have a couple of clicker questions, but I think for the sake of time, I really want to move into regulation of gene expression. But this is sort of what I do want to kind of just talk about this, just really quick. If you have a protein with an amino acid that is leucine, alanine, serine, and those have the mRNA code, what is the corresponding DNA template? So this is just asking you to go backwards. So basically this is just saying if you have RNA, what is the complementary DNA? So in order to do that, you have to know that RNA has U instead of T. So this would mean A, C, right? So that's going to be one of the threes, one of those, G, C, T, G, C, T. We already got it right here. All right. Well, let's see if I'm right. Thank goodness. Right. So this is just asking you to go remember, kind of like help you think about those complementary nucleotides. Remember complementary base pairing. And remember the difference in bases in DNA versus RNA. Very important. All right, and then which of the following involves a promoter region? Just a reminder that in genes, transcription has a promoter region, and the promoter region is where RNA polymerase binds to start the process. So if it's binding to start the process, it's initiating the process. And that's going to be transcription. All right, so let's move forward, shall we? I'm excited. I love talking about how gene expression is regulated. So you mentioned something about the Tata Box. I'm guessing you already started, good job. The Learn Smart Reading assignment for gene regulation, because that's where a Tata Box is involved. So we're going to talk about the Tata Box, and we're going to talk about some other ways that genes are regulated. I think this is just like the coolest stuff. Okay, so just for reference, these are the chapters covered in this. We're not going over the entirety of chapter 11 or 12, but just some select portions of it. Okay, so how are genes regulated? So basically what this slide is just talking about is, as you know, organisms are at their basis divided into prokaryotes and eukaryotes. So remember, prokaryotes are primarily single-celled organisms, but what distinguishes them is they do not have membrane-bound organelles, including a nucleus. So this says bacteria. Prokaryotes have a lot of ways of regulating their genes, but primarily they regulate their genes using what are called operands. So we're not going to go into operands, unfortunately. They're really awesome, but you will get them in other classes if you take micro, or I think actually in 182 you'll get them as well. They're really cool, sort of simplified regulatory systems. Basically, a bacteria will have all of the genes necessary to like use milk sugar as an energy source, as opposed to glucose using lactose. Like, it has the genes to do that, but basically they're always off, and they only turn on the genes they need if they're like in the presence of it. So they have the genes to use lactose for energy, but normally those genes are off. But if they see lactose, it's lactose itself that turns on those genes. So that's like an operand kind of structure for all bacteria. Most prokaryotes, what they do is it's environmental signals like the presence of a sugar that allows them to turn on genes to use that sugar. It's really cool. But that is their primary method of genetic regulation. They're very simplified. Eukaryotes tend to be much more complex organisms. Not to say their genomes are larger necessarily, but they do tend to be far more complex. They have a lot more genes to regulate, and they have a lot more environments that are more complex. Like bacteria, for example, E. coli pretty much only likes to hang out in your gut. And your gut, throughout your lifetime, it changes. It does change, but it doesn't change that drastically, right? So you're not going to go from like loving cheeseburgers to hating cheeseburgers. Your bacteria are not going to be like, oh my God, no more cheeseburgers. If they do, I'm, okay. But that drastic change, right? You're not going to switch from cheeseburgers to like only kale. You're going to switch from like cheeseburgers to like black bean burgers, right? You make a switch to vegetarian. It's not crazy drastic as far as nutrient content in the gut. And that's where E. coli live, right? So they don't have to regulate their genes like too much because their environment tends to stay pretty much the same. So this is not like us, right? For example, me, I was born and raised in Missouri, and then we moved to mid-Missouri, which is surprisingly different as far as temperature and climate. And then we moved to Seattle, and then we moved from Seattle to Arizona. So my body, the bacteria on my body, I've put them through the ringer. My body has also been put through the ringer because of this. Because of that, my body has to be able to adapt and change based on all that. Right? Like, I need to sweat more in Arizona. I need to turn on the genes to sweat. So as eukaryotes are more complex, their environments are also more complex. So they have to have a lot more ways to kind of like turn on and off their genes or regulate their bodies, regulate transcription and translation basically. So you can see here under eukaryotes, we have a lot of different ways of regulating gene expression. We can, and we're going to go through these, but we can basically like change DNA structure. We can change how mRNA is spliced and kind of decide how long RNA stays around. Transcription factors are the primary method that are used by eukaryotes, huge. We can also do stuff that's post-translational, which is modify proteins. So eukaryotes compared to prokaryotes, just tons of different ways of regulating their gene expression, which is very important. And then I do want to point out sort of at the top of the slide, I just say like all, because what NC equals non-coding RNA, we're going to talk about some non-coding RNAs. But I do want to mention that non-coding RNAs that are regulatory are present in both eukaryotes and prokaryotes. So all organisms that we know about utilize non-coding RNA for gene regulation, whereas operands are only present in prokaryotes. And then to our knowledge, these methods of gene regulation are primarily present in eukaryotes. So they're kind of more restricted. But non-coding RNA are in both prokaryotes and eukaryotes. Okay, so what this slide is showing, which I think is pretty important, is kind of, well, first off, let's take a look at this slide and then you'll understand. So at the basis, it's showing a eukaryotic organism. So that's primarily today, we're going to be talking about gene regulation in eukaryotes. So all of this, right here is the nucleus or the nuclear membrane. So all of this occurs in the nucleus. Remember, DNA is in the nucleus. And so in the nucleus, DNA is transcribed into mRNA. So most mRNA processing also occurs in the nucleus. So we go from like pre-RNA, which that's what it means by RNA transcript, is pre-mRNA, which is basically that pre-processed. And then we process it into mRNA. So that all occurs in the nucleus. So that's why we have that nuclear membrane here. Then that mRNA leaves the nucleus, enters the cytosol, and associates with a ribosome for translation. So that's all occurring in the cytosol. You can see we go from mRNA to protein. So that would be number 4, would basically be a ribosome. Let's go ahead and put that there. Would be occurring at a ribosome right here. I don't know how to, there we go, where we make a protein. But then we can also have that protein, you know, do stuff. So that's what you're looking at here, the process of going from DNA to RNA and its processing, that RNA leaving the nucleus, and then associating with a ribosome for translation. So when we talk about where, at what points during the process of gene expression can we regulate this? Like, and what we mean when we say regulate it is, you know, we may start to transcribe a gene or we may start to transcribe a gene to become a protein, right? Let's say that we just chugged a gallon of milk. So we need to be able to like digest lactose. Right? So we need to turn on those genes. That is very important. But let's say it's like 2 hours after we don't chug a gallon of milk. That will kill you. But let's say it's like 2 hours after you drink a glass of milk. Your body doesn't necessarily need to be like going into overdrive to be making the enzyme that breaks down that milk sugar. So it can kind of slow that down. So what we talk about when we talk about gene regulation is basically deciding when and at what level, like how much we're making protein. So we can ask ourselves, do we need this protein? Yes or no. How much of this protein do we need? A lot or a little? Do we need this protein all the time? Right? So those are the questions we can ask. And what gene regulation allows us to do is basically decide when a gene is transcribed, right? So when are we going to actually make a protein? So if you remember what I said, when I said the big difference between replication and transcription is that whenever we go through replication, which is making a duplication of a DNA strand, right, for the purposes of reproduction. With replication, you take the entire genome, all of the DNA, and you make a copy of it all at once, right? But a big difference is that in transcription, we have our DNA organized into genes, so those units that code for proteins. And with transcription, we don't transcribe the entire genome all at once. The reason we don't do the entire genome all at once is because we don't need all of our proteins all the time. We only need them basically circumstantially. We only need lactase when we are actively drinking milk. Why waste the energy to make that enzyme unless we need it right then, right? So that's what gene regulation allows us to do, is basically decide what genes are expressed and when and how much. So you can see on this slide that there are lots of points, because this is such a very important thing to regulate. There are lots of points at which we can regulate this. So for example, I have this arrow here. I really don't like in science saying like the most important, but I believe that that's what your textbook uses. So I tried to be consistent there. But I really don't like saying that because all of these are very important. But regardless, probably how could I rephrase that? One of the most, basically, if it gets past step 1, it's very likely that a protein will be made. If you make RNA, right, pre-mRNA, if your gene gets transcribed, basically, it's pretty likely it's going to be all the way translated to a protein, is what that means. It's not necessarily the most important step of gene expression, but it's just like, it's sort of a threshold that if you get past step 1, you're probably going to get all the way to a protein. So it's sort of one of those really important checkpoints. I had a question. All right. So do genes have memory, or can they forget their function if they stop their function? For example, vegans decide to not consume animal products. Can they at some point go back to eating normal and not be able to digest those products? That's an excellent, excellent question. What I'm going to do is copy that question, and I'm going to paste it in this for later, because we will get to a point where I'm going to be able to better answer that question after we talk a little bit more about gene regulation. So let me come back to that question. Great question. Let me come back to it. Okay, so with where can gene expression be regulated? So we have tons of different steps throughout the process of transcription and translation, where we basically can say, yeah, let's go through with this, or hold off, let's hold off. We can, number 1 is saying we can basically stop this process when we are actively transcribing DNA into mRNA. So we can stop that. As you can see, that's kind of one of the biggest thresholds. Once we get that mRNA, we can stop processing. So we have that pre-mRNA. We can basically prevent it. If we don't want to make the protein, we can prevent it from being capped or tailed or spliced, etc. And if we prevent that, it won't be able to leave the nucleus, associate with a ribosome. And if we get past that, we have mRNA, and what we can do is we just can prevent it from exiting the nucleus. If it can't get out of the nucleus, it's not going to be able to associate with a ribosome, so it won't be translated. If it does escape the nucleus, what we can do, you can see sort of RNA transport localization. We can prevent it from getting to a ribosome. And then a couple of important things that we'll talk about more specifically later is we can actually interrupt the process of making that mRNA into a protein. One thing we'll talk about, we do have methods to just like destroy mRNA when it's in the cytoplasm, which would prevent it from being translated. But we can also actively interrupt the process of translation. So interrupt ribosome function. Let's say we get through all of these processes and then we finally have a protein. We actually do have a lot of regulation of protein as well. So for example, the vast majority of your proteins are created in an inactive state. So they're created proteins in an inactive state, meaning they don't work. They're off and you have to turn them on. So even if you get all the way through this process, we can regulate it then. So you can see here, this is just showing 7, but this is just a summary of all the ways that we can regulate gene expression. And what you should always see is that if a biological process, such as transcription and translation, and what we'll talk about next, which is cellular respiration, if a biological process has this many steps where we can say, never mind, that should tell you that it is one of the most important processes in the body. And transcription and translation is, all right? We have to be able to say, go forward or don't proceed. Okay, so let's go through all of these, shall we? We're gonna start today. We definitely won't finish, but that's okay. It's fun to talk about. Let's take our time talking about it. Okay, so let's start from the beginning, right? So we can, we're gonna take a step back even from the step 1. So we can actually start to regulate gene expression, and we regulate gene expression all the way into how DNA is stored. So first off, it's important to note, we talked about this when we talked about replication, right? So we talked about the fact that our DNA is stored, wrapped and wrapped and wrapped and wrapped and wrapped and wrapped and wrapped around proteins. And these proteins are called histones. So you can see in this picture how DNA is stored. So we have our double-stranded DNA molecule, and it's a helical structure, which means it's kind of wound like that. And it's wound even further around these histones, which actually kind of grab the DNA and then wrap it around themselves. And then they wrap it even more. So we wrap our DNA around histones, and then those histones bundle and wrap it even further around those histones, and then that's wrapped even further and further and further. So this process, whenever DNA is wrapped and wrapped and wrapped and wrapped and wrapped, that's what we call being condensed, right? So DNA in its condensed form means that it's wrapped and wrapped and wrapped and wrapped and wrapped really tightly around those histones. So just a reminder that if we want to even start transcription and translation, right? If we want to do this process, we have to be able to take RNA polymerase, actually back up way back here. We have to be able to, where is it? Right here. No, right here. Right? If we want to be able to access a DNA strand for transcription, we have to be able to take a DNA strand and separate it enough to get RNA polymerase to be able to read that template strand into RNA. You have to open up DNA. In its condensed form, right, when it's wrapped and wrapped and wrapped and wrapped and wrapped around those histones, it is inaccessible to any enzyme, right? So if you have it condensed into a chromosome or in its condensed form, what we call chromatin, it is inaccessible to enzymes. So this includes DNA polymerase for replication and RNA polymerase for transcription. So if you remember, kind of just revisiting this, if you remember the process of mitosis and meiosis, remember the big thing that happens during interphase compared to prophase is during interphase, your DNA is actually kind of like dispersed in the nucleus, right? And what we mean by that DNA being dispersed is it's not condensed into chromosomes. So during interphase, which is, if you remember from that lab where we calculate the lengths of time for each of the different phases, if you remember, interphase is the time that a cell spends the most of its life in. Cells spend the majority of their time in interphase. And that's because a cell can only be sort of active transcriptionally when DNA is not completely condensed, right? If we condense our DNA into chromosomes, which happens during prophase, which we only enter prophase if we are actively going to duplicate. If the only thing we're going to do is go from one cell to two cells, only then do we enter prophase. If a cell is just functioning normally, it's going to be an interphase. And the reason that is different is because during interphase, the DNA is accessible. It can become transcriptionally active compared to during prophase when you have that condensation and the DNA is so inaccessible because it's so tightly wound. So even though that's kind of the drastic example, condensation into chromosomes, even during interphase, your DNA is still kind of wrapped tightly to protect itself. But it can just be unwound and rewound a little bit easier. So here, how we can actually make DNA accessible to RNA polymerase for transcription is via a process called acetylation. So something to note is that how we get DNA to wrap around this histones is via the process of methylation. So remember that proteins are made of amino acids and histones are proteins. So that means that these histones are composed of tons of amino acids. So for methylation, what happens is we have a methyl group, which a methyl group is a carbon and 3 hydrogens. We have a methyl group added to amino acids of the protein structure of those histones. And when that happens, it causes the DNA to wrap a little bit more tightly around those histones. So we can add or remove those methyl groups on the histones, and we do that with an enzyme called a methylase. So if we need to take DNA and condense it into chromosomes, a methylase will go through and add methyl groups to those histones, and it'll cause the DNA to wrap tightly. So that's how we condense it. But let's say we want to decondense that chromatin, open it up so where it's more accessible to those enzymes, to RNA polymerase for transcription. If we want to open up, we need to remove those acetyl groups. So in this case, what we do is we add an acetyl group, which is basically 2 carbons, an oxygen, and 3 hydrogens. And in order to do this, we use an enzyme called acetylase. And it basically just replaces those methyl groups with that acetyl group. But what happens is DNA doesn't like acetyl groups as much as it likes methyl groups. So if you start adding those acetyl groups or acetylating that those histones, DNA starts to unwind. Okay, so I want to really specify something, and this is something really important, is that methylation and acetylation of histones, specifically of histones, is what allows DNA to basically condense and decondense to allow DNA to become transcriptionally active, right? So we will talk later again about another type of methylation. That's why I'm specifying this. We will talk again about another type of methylation because you can methylate a lot. It's pretty easy. But this type of methylation, when you're methylating specifically those histones, is what allows DNA to either condense or decondense. And again, if you want DNA to be transcriptionally inactive, you would use methylation because it wraps the DNA really tightly and therefore those enzymes can't access it. If you want DNA to be transcriptionally active, meaning we can transcribe it into proteins, then we have to open it up, right? Make it accessible to those enzymes. And that would be acetylation, specifically of the histones, not of the actual DNA, histones, right? So we actually have terms for DNA when it's transcriptionally active or inactive due to methylation or acetylation. And you can kind of see the difference here. So this is looking again, the black is going to be your DNA and the blue are your histones. So if you have, basically your DNA is wrapped really, really, really tightly around those histones, or very, very condensed, that's what you call heterochromatin. And heterochromatin is sometimes referred to as like silent or silenced regions of DNA. It's so condensed around those chromosomes, around those histones, that basically our enzymes can't access that DNA, and so they can't transcribe it. So if we were to look chemically at heterochromatin, we would see lots of methyl groups here, all right? Because again, methylation causes DNA to wrap tightly around those histones, therefore silencing it. If we have transcriptionally active DNA, this is what we call euchromatin, and that's what we mean by active. And if we have euchromatin, we would have less methyl groups and more of those acetyl groups. Oh wait, I forgot. COCH3? Yeah. Why does that look so wrong to me? Anyway, okay. Well, you get the point here, is that those are subscripts. Okay, so that's kind of a difference between transcriptionally active DNA or euchromatin, is that it's active, can be made into proteins. Our enzymes can access those DNA strands, whereas heterochromatin is silenced, and that's because it can be accessed. Our important enzymes can't access that DNA, and that's just a lot of methyl groups. All right. Hopefully that makes sense. And again, I really, really want to highlight the fact that when we're talking about condensation of the DNA, so euchromatin or heterochromatin, what we're talking about is we're talking about methylating the histones. All right, so how we're wrapping that DNA. All right. I mentioned that because there is another thing that we can methylate. And here we are talking about methylation of individual bases. And in this case, we're talking about those nucleotide bases, right? So for example, you can have methylation of thymine or, guanine or cytosine. Usually methylation takes place on cytosine. A lot of C's get methylated, but it can happen to any nucleotide. So here's why this is important. What happens with methylation of histones is that causes changes in the DNA structure, right? It wraps tighter or it becomes looser, basically making it more accessible or less accessible for transcription. So once we get into the effect of methylation of individual nucleotides, what we're interfering with is the actual process of transcription. And that's because there is a big difference, right? There is a big difference between, like, let's say C as a nucleotide, so cytosine, and this is CH3, just backwards, right? So methylated cytosine. There's a big difference between those two. Essentially, your RNA polymerase can recognize cytosine, whereas it cannot recognize, well, just for clarity's sake, I don't know why I'm being so weird about that. Just RNA polymerase cannot recognize a methylated nucleotide. So what this means is that if you have more methyl groups on the nucleotides in a strand of DNA, RNA polymerase isn't going to be able to read that into mRNA. So we can look at a strand of RNA or DNA, and we can see if it has a lot of methyl groups present, that means RNA polymerase is not going to be able to read it, so that gene is off. Whereas if there are not a lot of methyl groups, RNA polymerase will be able to read it, and that gene can be on. So what causes methylation of individual nucleotides? And the answer is, we're not 100% sure. This is one of those newer kind of amazing things. But what I want to mention is that the reason we talk about this is this basically regulates, how does this, it causes phenotypic changes without altering the genotype. This is the biggest thing, the reason we talk about epigenetics is because historically we know, or the kind of rule was that genotype leads to phenotype. You have a gene, it's transcribed, translated into protein. The only way you would get an altered protein or kind of like alter that process is if you alter the nucleotides in a strand of DNA. So the only reason that you would get a change in phenotype is if you have a change in your genotype. So remember, the genotype is going to be the sequence of nucleotides, A, T, C's, and G's. With what is called epigenetic change. The epigenetic change, you do not get a change in nucleotide sequence. You still have A, T, C, G in a gene. But what happens is you get methylation, so just a slight chemical change in a nucleotide, and all of a sudden that can change the phenotype, the resulting phenotype. This is talking about a gene being on or off, but sometimes that change won't completely turn a gene on or off. Sometimes it'll just change, it kind of functions sort of like a genetic mutation. So, so you would still get a phenotype, but it would be different. So this kind of helps us to explain why there are certain disorders that you can't basically detect by doing genotypic analyses. You can't look at a person's genotype and say they're going to have this disorder. And it's because it's not a genotypic mutation. So what causes this? And I want to mention also that this is not only contributing to, it's not only contributing to like disorders. We're kind of like talking about it in the context of genetic disorders or epigenetic disorders. But there's actually a lot of really important regulatory things that happen via epigenetics. We don't really understand epigenetics fully. We do know that these changes or methylation, just like methylation is reversible on histones, methylation can be reversible on nucleotides as well. So we can add methyl groups or remove methyl groups. We know that our body has the capability of doing this. Why it does this is sort of unclear. We do know that just like genetic mutations, the environment can also influence epigenetic change. But what we do know is that this is one reason why, like you can see in this picture, so this would be like your grandma carrying your mother, and then your mother carrying you, and then you. So for example, there is a link between smoking and obesity. But interestingly enough, this is why, like, if your grandmother smokes, it can lead to you having obesity, even if your mom didn't smoke. Because what can happen is your grandmother smoking could lead to epigenetic changes. And those, as opposed to like genetic changes, which might occur, you know, in her baby, your mother, those epigenetic changes can happen in your mother, but they can also happen in the nucleotides, like let's say of your oocytes, your eggs. And that's why, you know, your cells can result in you having those mutations, but not your mother. So we can see that obesity kind of happening over time. So that's just one example. I think probably a really interesting example, but I think this is all very interesting. I think epigenetics in general are just like insanely interesting. But one reason I find this quite interesting is because I have a lot of twins in my life. In fact, my husband is not a twin, but he has two brothers that are twins, and then he has cousins that are twins, and like my grandmother was a twin. I mean, we just like for some reason have tons and tons of twins in our families, which is why, you know, right now I only have one child because I'm like terrified to have more. I might not go from 1 to 2. I might go from like 1 to 3. I don't know if I'm ready for that. But it's hypothesized that one of the reasons you can have like identical twins, right? Identical twins means that their genotype, their genes, their nucleotide sequence in their DNA is identical. One twin has ATCG, the other twin also has ATCG. But if you take two identical twins, I'm sure several of you know pairs of identical twins, they are very different from each other, right? So this kind of gets you into the conversation of like nature versus nurture. We know that if you take two identical twins, right, their genotypes are exactly the same, but they will become individual people. And nature versus nurture is the way it was referred to in the past. But you can say, for example, like my husband's brothers are identical twins, and they were raised, you know, exactly the same. But one of them actually has anger management issues and possibly like bipolar disorder, and the other doesn't have this issue. So what caused this one to have that, you know, phenotype and this one to not? And the answer is, again, their genotype is exactly the same, but something during their development would have caused like changes to, probably epigenetic changes, to the individual nucleotides. So their genotypes were not changed, but their phenotypes were. So this one is drastically different than this one. They also moved to separate areas. So one stayed in Arizona, one moved to Seattle, spent a lot of their times in different locations. So there's just so many things. So what we know is that these changes can be influenced by the environment. So epigenetic changes are kind of one of our kind of emerging explanations for that nature versus nurture hypothesis. I don't have time to talk about this all day, even though I would love to. So what I did is I posted some stuff in this PowerPoint to kind of go into it. These are some links to some really cool, this genetics, epigenetics is a really fun interactive activity you can do, and these are just 2 videos talking about epigenetics. I also suggest this book. I love pop culture, and this book kind of talks about epigenetics in the context of pop culture. So how can interacting with society and pop culture and watching TV and using our cell phones, how can that influence our phenotypes? So for example, if you look at human evolution, one thing that humans are evolving is, I have one, but maybe I was born with it. One thing that's happening to humans is they're kind of developing hunches. So little kind of like humps on their back, their necks are kind of pushed forward. We're more suited for like this position. And the reason is not because genetically we're designed, like posturally, we're, our genotype will suggest that we are supposed to have like excellent posture, right? We are built for excellent posture, but we look like this instead. And the reason is because this is how I'm doing this. But also, more importantly, this is what I'm doing. So we are actually having, like how we're interacting with our environment is directly influencing our phenotype without necessarily changing our genotype. So this talks about it, pretty cool stuff, that book, but yeah. Okay, so again, I want to mention the big thing about epigenetics that will come up, the way people refer to it and the way I would recommend you remembering it is that it is a way to get phenotypic change without alterations in the actual genotype. Okay, so that is all we have time for today. But I got a couple of questions. So I got questions about this excellent question, sort of memory, I want to get back to. We also had a really good question about the Tata Box. And I also had another amazing question, such as how radiation influences nucleotides. All super good questions, and I promise you that we will get to them. We just didn't get to address them today. But I noted them so we can address them. We will address them as we go. So, but that's all we have time for today. So with that, I will see you again on Thursday. We will continue this awesome lecture topic, and we will move into on hopefully Thursday, but probably next week more into genetic mutation. So yeah, and if you have questions, type them in the chat box or email me or type them on the discussion board, however you want to get to it. Ask them. All right. Have a wonderful day, and I will see you later. Stay safe. See ya.